Imagine a technology of the future, one that gives you the ability to create a heartbeat, or a technology that enables the ability to walk again. Imagine a technology of the future that can find the answers that give the gift of sight, and one that can develop personalized treatments for cancer patients. Imagine a technology of the future that is the reality today. That technology is bioprinting. From fabricating tissues and organs to testing the safety and effectiveness of new drugs, we are quickly approaching an age where what once seemed like science fiction is now a reality, where the possibilities of 3D printing are truly endless. But not without the efforts of the scientists and researchers pushing the limits of tissue engineering. Dr. Stephanie Willerth is a professor at the University of Victoria and stands at the forefront of the tissue engineering revolution. Her research is pushing the boundaries of what we thought possible, using 3D bioprinting to fabricate tissues and eventually organs that could one day transform healthcare. Imagine the impact of being able to print a new pancreas for a diabetic or test drugs on bioprinted tumors to ensure their safety and efficacy. Dr. Willerth's influence extends beyond her groundbreaking research in academia. As the former president of the Canadian Biomaterials Society and as the CEO of Axolotl Biosciences, she has been a driving force in advancing the tangible industry of biomaterial technologies, making strides that bring us closer to a future where personalized medical solutions are within reach. Today, Dr. Willerth joins me to delve into the remarkable progress in bioprinting and tissue engineering. We'll explore the crucial role of stem cells as a foundational material and discuss the significant challenges the industry is likely to encounter in the coming decade. Following is my discussion with Dr. Stephanie Willerth. Dr. Willerth, thank you so much for joining me today.
Oh yeah, no, I'm excited to be here.
Now, tissue engineering, regenerative medicine, bioprinting, these are really the sort of things that sci-fi dreams are made of, but they're very real, and you live in that world every single day. What is regenerative medicine? What is tissue engineering, and what is bioprinting?
Yeah, well, it's always funny because I think, although it's been a while, with Westworld, they always show them printing horses and humans using plastic, which I do actually have one of my 3D printed axolotls here, which is not actually how you make a human tissue. But regenerative medicine is the science of trying to make substitutes for diseased and damaged tissue and regenerate, the cells and organs that you find in your body. And tissue engineering goes along with that, where you're trying to figure out how you can combine things like cells, biomaterials, which are materials specifically designed to interact with your body, different types of drugs and drug delivery systems in order to heal the body. And then with bioprinting, just again going back to 3D printing, which is traditional. Here, when you're making an axolotl to sit on your desk, you heat up a filament, you extrude it, and then it goes into the shape that's in the CAD file that's specified. And bioprinting similar, and it is a subset of tissue engineering, but instead of a hard plastic filament, you want to use materials that are going to keep your cells alive and functioning in a manner similar to that found in the human body. And so the field's been probably around since the 90s, with 3D printing being around longer than that. But one of the big challenges was, how do you actually print cells while keeping them alive? And then the next big step was, okay, you've printed cells, but are they going to actually resemble something that you would find in the human body? And that's where the field has been taking some very exciting recent advances, where we're actually being able to replicate some of the functions you'll see in the human body.
Fantastic. I'm glad that you brought up the axolotl, the 3D printed little
Yeah.
guy there. I was going to ask you, what are the main differences between traditional 3D printing that's been around for a while now, that focuses on extrusion of plastics and polymers? What is the difference between that and what you're doing with live cells?
Yeah, you have a much smaller operating window when you're doing bioprinting, just because cells are alive, and in general, they don't like to be too hot or too cold. They're also really sensitive to shear, so the forces that you put on them when you're extruding them, whereas with plastics, it's really easy. They can extrude really nicely and then form together to make structures like the one I just showed you. Given that 3D printing's been around longer, you get a lot better resolution. While you've mainly mentioned plastics, there's actually been a lot more work being done in metal printing, which can go towards being orthopedics.
Yep.
Yeah, but I think, and then with bioprinting, the other big challenge is, if you want to do what my research group does and what other groups have been doing around the world, which is really one of the big advances, is you need to print millions of cells per mil, because our human tissues have millions of cells per centimeter cubed in our body. So being able to get that many cells and then also keep them alive, and when you put them through, because cells are typically, you typically grow them in a dish. They're pretty finicky. You have to grow them at physiologic conditions. So, 37 degrees C, a certain CO2 percentage. So keeping your cells happy can be difficult in the best of times. And then essentially sitting through the roller coaster that is a 3D printer, they aren't always the happiest, especially what we work with, stem cells. So it's been a challenge finding bioprinters that are sophisticated enough to handle them. And then also what my research group does is work a lot on the materials that you surround these cells with so that they actually do survive the printing process.
So is it safe to say that 3D printing is sort of the next generation of tissue culture? You're doing sort of like constructive tissue culture. You print what you need to print, and then you go ahead and treat it like any other tissue culture. But you've brought it to the next level, essentially, with this technique.
Yeah, and I think although for my research group, which focuses a lot on neural, and I always feel like I'm tempting fate when I say this, you aren't going to or you should not, especially be printing. I mean, we print regions of brain tissue and you can see Alzheimer's signals and things like that. But at the end of the day, you won't be implanting into a brain a 3D printed tissue. I can see, and people do this, like very specific cell populations, for example, dopaminergic neurons to treat Parkinson's. But just like given the nature of the brain and how important it is in terms of function, you'll probably be injecting cells rather than printing a specific region. And also, if you think about things like traumatic brain injury, each one's different, or spinal cord injury. So it's not really something that lends itself as a regenerative technique for 3D printing to fix those types of defects. But if you look at things like the heart, it makes a lot of sense, because if you're going to make a replacement organ, and again, this requires lots of challenges to be met with cell sourcing, printing the appropriate cell spatially, ensuring the correct function. But you could scan a person who has a diseased heart to get its dimensions, which is very important. If you go back to when they initially made the first artificial hearts, they were actually so large you couldn't implant them into one. So, so the fact that you could scan a patient's heart and get those dimensions, and then in theory feed that file into your 3D printer, you could actually make a replacement heart, provided you solved all the cell sources. The same for bone defects. You can scan like those, and those things make a lot of sense to 3D print. The other one that I always think, although interestingly, it was a group out of Cornell with a company called 3D Biotherapeutics that did deformed ears that actually put 3D printed tissues into humans. But skin is another one I think makes a lot of sense. You have a burn victim and you can have a handheld printer, and then you can just directly print the skin on to people.
Right. Oh, there's so much that I want to unpack there.
Yeah.
So that's interesting, talking about scanning of the heart, or maybe even scanning of the bone, or scanning of any other organ that you want to print. Do you think that might be another thing that we can mediate to prevent rejection? You're using somebody else's cell or their own cells as source to prevent rejection if it's some sort of tissue you're implanting. But would it help to get the size of the heart right? I know it's probably not directly related to rejection, but it's a better fit, right? Yeah. In that body. So with a better outcome, essentially, at the end of the day. That's something that I've never thought of. That's very interesting.
Yeah. No, I think that's something that, it makes a lot of sense that if you can do that for that type of tissue, like hearts or lungs, getting them to size right. I mean, it was obviously a big scandal, but the doctor from the Caroline Institute that the tracheas that did not work based on fake data, like that was one of the huge issues with implanting them into kids, was like the tracheas weren't the right size, and like they couldn't fix them as they grew. And so I think that there's sort of two things there. One is like being able to customize it physically. And then the other part you touched on, especially with some of the stem cell technology, getting the cells to match. That's another thing that potentially, if you're doing 3D printing, you could make one of these stem cell lines from the patient, use that to make the cells you need to do a personalized type implant as well.
So let's talk a little bit about stem cells, because you've mentioned it a couple of times now. It sounds to me like stem cells are sort of a key to advancing the technology, so that you've got appropriate sourced cells from the person who's going to be receiving some sort of transplant at the end of the day, and so that you can make the tissue you need to make by forcing differentiation down the right path. So can you tell us a little bit about stem cells and then your iPSCs?
Yeah, for sure.
Induced pluripotent stem cells, right?
Yeah. So for people who aren't stem cell biologists, like the main two properties of a stem cell, and this is why we use them a lot for tissue engineering. One, they can become multiple cell types. So some things like mesenchymal stem cells can only become things like fat and bone and cartilage. The human-induced pluripotent stem cells we use, or embryonic stem cells, can form any cell type found in the body, which is why people always talk about them as a tool for tissue engineering, because in theory, then you can make any tissue. The human-induced pluripotent stem cells, which were discovered when I was in graduate school, they're really exciting because at the time Yamanaka took a skin cell and they treated it with factors and then went back to behaving like a stem cell, which is great because then we could all have our own stem cell lines. Because for most of us, there was one embryo and that's what made us. So we aren't getting our own embryonic stem cell lines. So that was something I was really excited about. One of the reasons we work with them, although there is also, I know, really good work being done out of the University of Toronto and Andreas Nagy's group. And actually one of my friends, Evan Scott, who's moving to the University of Virginia. So Andreas's group makes stem cells that don't have immune markers on them. So then so they're universal stem cell. So like if you made a heart from them and put them into someone, the body wouldn't recognize them as foreign because of how the cells have been manipulated. So normally your cells, yes, normally all of our cells have two different types of immune molecules. One is a more broad MHC complex, which I think there's like 6 specific groups worldwide, which usually corresponds to ethnic groups. And then there's more specific ones that go for each individual human. And so these are some of the reasons why you get rejection when you implant organs. And so, as I was saying, the idea of this is you engineer the stem cells so they don't express these molecules. And so then you don't get rejection. And then what I was going to say about what's interesting about my friend Evan Scott's work, Dr. Scott, is they've actually made, he does immunoengineering, again, manipulating the immune system, because this is a big issue. They've made particles that will coat the cells you're transplanting. So then your body doesn't see those molecules. So in this case, you're transplanting still patient-specific ones, but because you have these nanoparticle therapies, your body doesn't see them as foreign and then doesn't reject them. And so that's one of the big, I'd say, so there's 3D printing and its challenges, which again are always like scale up, amount of cells.
Right.
And interestingly, if you want to print organs, it is really a manufacturing process. So like even the printers we have in lab, we're using on a good day, 50 mils of bioink, which is getting you little, so we print like miniature brains that are half domes. So they're like a centimeter in diameter and half a centimeter high. So you might get like 100 of these. But if you actually wanted to print, think about a liver, which is much larger, you're going to need.
Sure.
My friends who have bioprinting companies, this is something they've addressed. They have a huge vat of bioink, and sometimes you need mixers to make sure everything's consistent because, you're going to be printing for a while when you're printing the larger things, organs.
Right. So it sounds like what your colleagues are doing there, and you said the word scale up, one of the challenges going forward into the near future and long future is having a reliable source of cells. So being able to scale up that source by doing some of the engineering you referred to, I guess would go a long way into solving that problem. I'm happy you brought up the term bioink. Can you define that for us?
Yeah. So if you think about traditional 3D printing, which I think is pretty common these days, which is, normally extruding a plastic filament. So I just say in the 3D bioprinting process, the bioink is our equivalent of the filament. And usually for bioprinting, you have your cells seated inside. So it's kind of a living filament that you're printing. And usually you're printing something like a hydrogel. So for normal people, just think something like Jell-O. The consistency of Jell-O is usually, because it's softer and more protective of the cells, is what you're doing. So essentially the filament you use when printing a living tissue.
Another term that you brought up earlier was resolution. How do you define resolution when we're talking about tissue printing? With normal printing or 3D printing, we're talking about dots per square inch, the shortest point distance between 2 points that you can resolve. But in tissue engineering, what is resolution?
Well, it's actually the same for 3D printing and tissue printing, which is the size of the filament you're extruding, right? So like
Gotcha.
it's, I think we're like at 500 microns. So that's like when you actually have your printer like depositing the material down on your base and into your structure, like how big is your filament? And then
Understood.
Right. And so then that determines like how many cells you print. And then, if you're thinking about, as we always do, neural, right? So you think about the brain and all of its complexity, like what's the smallest region, like how many cells are in that region. And so that's sort of like what you can dispense in a certain area.
Now, when you're doing the 3D printing, laying down tissue, when you're using stem cells, are you doing the differentiation prior to the printing or after they've been printed?
We actually do both. And
Okay.
it's really interesting because when we first got our 3D printer with Aspect Biosystems, who shout out to them, they just signed, or they just were awarded, I think, $70 million to help
Hmm.
advance their tissue therapeutics. We actually printed
Yep.
everything at all stages. And it was very interesting because we found undifferentiated human induced pluripotent stem cells. So when they're in the state when they can become anything, we're actually very fragile and didn't want to print that well.
Mm.
We also print neural progenitors, which have been differentiated for about 15 days, and then they only give rise to the cells you'd find in the brain and spinal cord. And those print a lot better, and they were a lot happier in our ink and had better viability. And interestingly, we also printed
Interesting.
motor neurons, which were very mature. So they'd been cultured for 60 days prior to printing. And those were also very easy to print and actually had electrical activity
Mm.
one day post printing. So it was very
Interesting.
interesting when you work with the stem cells that, like, the stage of differentiation does affect printing and viability and function. And it really just depends on what you want to do. I do know in the cell stem cell paper from Zhang's group out of Wisconsin that I wrote a commentary on back in February, they actually were printing mature neurons and mature astrocytes, and they got really lovely results. So it's kind of interesting, the spectrum of what I thought would be easy to print. I really thought the ones that weren't differentiated would be really happy, but it turned out like some differentiation seems to really be important. And I think it also goes towards what you want your final application to be. If you're using them for drug screening,
Right.
yeah,
So it sounds like it really depends on every cell has its own hardiness or finickiness when it comes to printing. So that gradient needs to be explored and sort of solved.
yeah, and also like it depends on what you want. Like one of the reasons we'll print neural progenitors is because the neurons do connect and send signals to each other, and so it's helpful to have them in that 3D environment when they establish their
Right, right,
networks.
right.
Yeah.
So can you bring us up to speed as to what is the current state, the pushing the envelope of bioprinting? What's sort of the on the horizon right now? What's the coolest stuff that's being done?
Yeah, so I was very excited, as I said, when 3D Biotherapeutics actually did put
Mhm.
bioprinted ears with cells into humans, which I think was now 2 summers ago. I think Aspect has made some really interesting progress with their artificial pancreas for treating diabetes, and I also believe they have a liver program as well. And so they've been doing really well in terms of getting some of the preclinical data. And actually, the other big thing, because it is a new field, is building facilities and GMP facilities, good manufacturing practice facilities. So everything is kept track of for making
Right, right, right.
tissues to put into people, because we haven't really done that before. And I think
Yeah.
that's one whole area, as I mentioned, with our neural tissues. And this is something I know is going into the new hospital, I believe at St. Paul's in Vancouver, is they're going to have a 3D printing suite. And so there's also the whole drug screening aspect, which is something we focus a lot on, where we actually can take we can make these HIPSC lines from our collaborators' Alzheimer's patient, and you can print them, and we're hoping to test drugs on them. And then if we can see a drug that would reverse these Alzheimer's symptoms in a dish, you could prescribe it back to the patient. And while it's interesting because with Alzheimer's, so you can imagine it takes a while to age the tissues because you have to age them, and then as
Right.
they age, they get their symptoms. But our cancer projects, like for example, we grow glioblastoma cells in our bioprinted constructs, and they grow really quickly because our ink really does mimic the brain. And so the tumor's like, hey, we're home. And so I think that that's something where you'll see it, like, so you could, once the scale up issues addressed, I think in the future you'll see someone biopsy a tumor, the doctor will biopsy your tumor, they'll make a bunch of your tumor cells, they'll print a bunch of mini tissues with these tumors in them and hit them with the panel of potential cancer drugs and see what your tumor responds to, and then that'll go back to inform your medicine. And so I think that that's something that's also really, really powerful about the technology.
Yeah, I think what most people think of when they hear about printing or regenerative medicine is the transplantation aspect, but there is the drug discovery and the disease model aspect of 3D printing and tissue engineering that is maybe even more powerful at the end of the day because we have the ability to prevent disease with this sort of technology.
Yeah, and not to blow your mind, but this is something someone brought up when I
Oh,
was
please.
giving a public talk. So when we do take these cells from patients with Alzheimer's who are in their 60s, 70s, 80s, and we do see these Alzheimer's symptoms, someone actually asked me what would happen if you took cells from someone in their 40s, because again, these are stem cells and we accelerate the aging process.
Mhm.
Like, could we predict if someone is going to get Alzheimer's?
Oh,
Like,
interesting.
because we could take their cells and culture them probably for longer than the 60 days for the people who have it. But like, I was like, that's really interesting. I don't know how to write a grant to get that, but like, it was definitely, it's something I think about a lot, was like, because we can accelerate
Hmm.
aging in a dish, could we use it as a predictive tool to see if, because I mean, if you're 40 and, it takes,
Yeah.
you could potentially see Alzheimer's symptoms in 8 months and you'd know, like, would you start preventing things? I know they do that sometimes with genetic testing and people who have mutations, but I was like, in this case, you could actually potentially see phenotype.
Yeah, that's very interesting. That's fascinating. You blew my mind. That is a very interesting prospect, for sure. Obviously, if there's a correlation between what you see after you've aged your cells, it'd be hard because those would be very long-term studies, right? You'd age your cells, then have to wait 20 years to see if that person then sort of lands in what you've predicted.
Yeah. Well, the
So they
one,
would be long-term.
yeah, the other interesting one that came up is I was talking to a group of cancer doctors in Kansas City. I'm originally from Missouri, and they actually had a CAR therapy, so a cell therapy for treating cancer that worked in these patients and is now a standard of care. And they actually have all the patient tumor samples. And I was like, it'd be really interesting to print your patient tumor samples in 3D, treat it with the CAR, and see, because in this case, they actually know who the therapy worked for because they finished the clinical trial and it was successful.
Right.
And I was like, it'd
Right.
be really interesting to see how like our
Mm.
3D printed tissues would compare,
Yeah.
like, because we actually know the outcome. So that's another really interesting study.
Yeah, it's almost looking at it in the reverse direction. That's
Yeah.
extremely valuable data there. That would be nice. Yeah, you could write a grant for that one.
Yeah, I know. Yeah, we've talked about it. Yeah, there's like the whole Canada and NIH thing, but it's definitely, it was one of those things where I saw that data set and I was like, wow, that would be like really, really cool because the outcome. So
One of the things that crosses my mind when I think of the future of 3D bioprinting, where you're printing organs that are layered of tissues and cells that are very different and very specific. How do you go about um, printing a larger structure with many different types of cells and then having them integrate the way they would in situ?
yeah, well, so that's why that cell stem cell paper I was mentioning was really interesting. Is because they
Yes.
were printing layers of astrocytes and layers of neurons and getting really good integration. It does take a lot of playing around with cell numbers
Hmm.
and cell density. Um, and then, you have to also, with that, they also had really lovely electrophysiology, because that's like another thing that's really hard in a 3D tissue, is to make sure that, especially with like neurons, I guess with cardiac, you have to measure this electrical activity. I guess with cardiac, you can see them beating and you'll know whether it's beating like in a physiologically relevant manner or in an aberrant manner. And so those are some of the things you really have to monitor when you're like laying your cells to make sure that you're getting the proper functionality. And then it's interesting because we've almost had to go back. Well, I guess it is going back old school, because when we print our tissues, when we print them this densely, right? It's like when you do animal work, where essentially we are embedding them and doing histology, to look at morphology,
Right.
because essentially we've made tissues. And so as opposed to some of like the 2D cultures, or even some of our 3D tissues we made with more sparse levels of cells where, they're really easy to dissociate, really easy to stain. So doing some of the functionality is definitely another challenge. And then also, because it is expensive to print, you want to try and do this stuff in a non-destructive manner. So being able to
Right.
say continuously monitor your electrical activity over time is really, really helpful, as opposed to, having to do some of the electrophysiological stuff that is destructive testing.
I have a question that I want to ask, but I'm not quite sure how to phrase it. So let me see if this makes sense. When you are working with stem cells and you're printing them, and then you're forcing them to differentiate with factors like Yamanaka factors, or the whole suite that you're using now, how much of the development of those cells into functional tissue, the development and differentiation, how much of that is relying on genetics, and how much of that is relying on the intervention by the scientist?
Yes.
Like after you've printed it and then you force a differentiation, do you just sit back and let genetics take over?
Yeah, so we actually culture, so we kind of do both. So one is we start with usually okay, so this is for printing, we'll say our brain tissue. We'll start with these neural progenitors, they're restricted. So we're only going to be getting neurons, oligodendrocytes, and astrocytes. And then we actually have smart versions of our bioink. So have these little drug releasing particles that encourage the cells to become, usually for brain, if we're doing Parkinson's, we would usually want to make dopaminergic neurons, which are the ones that die off in Parkinson's. So those are phenotypically relevant. And for Alzheimer's, we usually make cholinergic neurons. So we have factors that are being delivered by the ink. We also use special media in order to ensure and promote that they'll be able to transmit their electrical signals. So I think it's a combination of both. You can use genetically modified cells and drive it in that way as well. The other interesting thing, although you didn't ask, is just something I think is kind of funny when you actually make the tissues, is for people who haven't worked with cells, if you just like look at them under a microscope, everything looks kind of clear. And even like our drug releasing particles look like little dots. And so for our tumors studies, we actually use GFP labeled cells, because then you can actually monitor the tumors with your microscope without having to stain them. And so that's something that a lot of groups use, is like fluorescently labeled cells, so you know where they are in your tissue. And also if you have different types of cells, again, a lot of them look really similar. So sometimes people, for example, when they're co-printing the neurons in astrocytes, they'll have like the astrocytes labeled, permanently green. They'll continuously express green fluorescent protein, and then the neurons red, so they know where they're going. And then it's also helpful because, again, you don't have to destroy the tissue to see if they're interacting.
That's a great segue, because I was going to ask you, my next question was, after you've printed tissue, you allow it to grow up. Let's say you like you said, you do a lot of histology. Can you tell the difference between a engineered tissue on a cross section under a microscope and something that is natural, for lack of a better sense, minus the GFP staining, if there is some of that? Just organization and structure.
Yeah, it really depends. Like some things look really similar. Like I think our tumors look pretty similar to what you'd see in an in vivo setting. Our neural tissues aren't as well organized, nor are they as complex as what you would find in the brain, although we do see electrical signaling in them. We do see, example with Alzheimer's, we do see tau secreted, and you'll see some of the tangles. So we are getting some phenotypic relevance out. Ideally in the future, we'll get more towards, printing more complex structures as well. Also, though, with the brain, it gets a little bit tricky. Well, because there is, at a certain point, you do see electrical activity. Some people have thought with like the miniature organoids, which aren't necessarily 3D printed, but you can get these miniature spheroid brain tissues that they're seeing brain waves. And so there are questions of like, how large should you make your organoids?
Sure.
And would you know if they were thinking, which I'm somewhere in the middle on, because I'm like, okay, well, you do see like these markers that we know are similar to brain waves, but like how would you know? Like your tissue can't talk to you, so how would you know? And it gets sort of very philosophical. It's like, what is thought, and how would we know?
Isn't it interesting that that's sort of a similar problem that's popping up with artificial intelligence at the same time? Both technologies have come to this point where you have the same question, whether something is sentient or able to think. That's very interesting. Now, what is the easiest tissue to print today?
I would say our lab has had really good luck printing skin tissue. The fibroblasts seem to really like our ink. It's relatively straightforward, and we're using adult cells that tend to grow relatively well. So they tend to be skins like, well, yeah, if you're printing just like fibroblasts and keratinocytes, it's pretty straightforward. We're also working with another microbiologist, Dr. Stephen Tufts here at UVic, to make them more complex, because real skin tissue also has a lot of bacteria that are both positive and negative, but healthy skin has, a whole microbiome working on it. And then when you have injury, then that's where your microbiome can get out of whack, and then you get infections. So we're trying to actually add in, although for those of you who've done cell culture, you don't usually want bacteria in your cultures.
Right.
But yeah, so it's interesting with that, to use that as a way to actually more accurately study wound healing. And that's not necessarily what we're doing, but I know some other groups have proposed doing this, like using it to bioprint models of the gut, and then again, see how the bacteria are affecting, because again, there's good bacteria and bad bacteria. And actually, I think there's been a lot of evidence. I'm sure everyone's heard of like microbiomes and like, people getting their
Of course.
microbiomes characterized, like how we can better understand that to improve health.
And in your experience, the most difficult tissue to print?
I mean, I think, as I said, neural is one of the trickier ones. I think neural and cardiac, especially if you're trying to get the different regions, are difficult, just because you also have to have the electrical stuff as well as structural components when you're lining up your cells, depending on like what region of the brain you're printing. And then cardiac also is very similar, where you have so many different cell types present that replicating all those features, especially if you want to make something for transplantation, is difficult. But I do know both a group out of Stanford led by Mark Schuyler Scott is doing an ARPA-H project, which is in theory building a human heart. And they've been doing a lot of work to find out how you'd make all these different cell types from stem cells. And there is a similar project being led by Dr. Lacron out of U Ottawa, and some other people, I believe, out of U of T, that are doing something similar, looking at how you would bioprint hearts. And again, because both these tissues have, the electrical and structural components, makes it a bit more difficult. And then, trying to get everything arranged and interacting is difficult. And then probably somewhere in the middle is something like the pancreas and the insulin secreting cells that companies like Aspect are looking at, which in that case, it's kind of interesting because you just need the insulin to be secreted. So there you aren't necessarily like your heart has to go where the heart goes. Whereas
yes, exactly.
Whereas with some of the diabetes treatment, they have outside of the bioprinting, it's like a credit card that they put the cells in, and then the credit card's made from a credit card shaped device that's made from material that your body won't attack as foreign. So, and that's something similar that they're trying to replicate with the 3D printing, is making a coating so the body doesn't see the cells as foreign. So that's somewhere in between, where you do need to keep your cells alive and functioning, but you don't necessarily need to transplant them into a particular location.
Okay, so that's what I was going to ask. So you're suggesting that with something like the pancreas, unlike the heart, that doesn't necessarily need to be transplanted into the proper location where you would find a pancreas, as long as it does what it needs to do, in this case, secreting the insulin.
Yeah. And so that gives you a bit more.
That's fascinating.
Yeah.
That is fascinating. So we've come a long way from the 1990s with the Vacanti mouse, which most people would recognize if they saw it in a picture. The chondrocytes from a cow that were grown on the back of a mouse that looked like a human ear.
Yep.
Are there any therapeutic therapies, I guess is the term. Are there any therapies right now that are being practically applied in hospitals when it comes to 3D printing of tissue?
Yeah. That's what I was alluding to earlier with the ear for microbiota, with 3D biotherapy. Right. Which is funny, since you brought up the mouse with the ear on the back.
Yes.
And then they actually did do ears. And the interesting thing about that, because, in my group, we started as academics. Now we have a spinoff company, but our spinoff company isn't on the clinical side, like companies like Aspect are. But with 3D biotherapeutics, I was curious because, as I said, I really thought skin, because there's a lot of reasons why I thought skin would be the first one to go clinical. It's easy to teach. If something goes really wrong, you can just take it off and put on a different skin substitute. Whereas, if you're bioprinting a heart and putting it into someone, it better work really well. But with those, what it turned out with the people with the deformed ears, it was interesting because the treatment is plastic surgeons doing reconstruction. And so by being able to scan their healthy ear and use that as the template to print the ear, it saved so much surgeon time that it made sense from the hospital billing, and this was in the US, it made sense from the hospital billing perspective that it saved over 50% of the cost. So it made a lot of sense financially to 3D print the ears as opposed to having the surgeons do it. And so that's something that's kind of an interesting driver of what goes in therapeutically. And I also think now that it's happened and it didn't have any ill effects and people tolerated it, I think it'll be interesting to see what therapeutics come next. I know for some of the stuff Aspect's been working on, because there's such a huge cell therapy component, that I think Health Canada is treating some of their engineered tissues as both a device and a cell therapy. So there's a lot of regulatory. And I do know that both Health Canada and the US actually have been working to try and improve the legislation to make it easier for 3D printed therapeutics. And both both therapeutics and, for example, applications for drug screening to become more accepted as technology, especially on the drug screening side, as there's a huge push to get away from animal models, which makes a lot of sense. I mean, mice don't get Alzheimer's or Parkinson's, and so we're using relatively artificial models of those
Right.
diseases.
Now, what I love about biotech for the most part is that it's extremely interdisciplinary. I think with the industry that you're working in, it's couldn't be more true. You've got cell biologists, molecular biologists, material science, biomedical surgeons, histology, microscopy, material sciences. The list goes on and on. So that I found really really cool, and I admire that about your industry. So which is great in terms of students and prospective careers. You don't necessarily need to be a cell biologist to get into the industry of tissue engineering, regenerative medicine. You can be a material scientist that does nothing in regards to biology, but is a expert in that. Um, what I wanted to ask you is, do you see, we're talking about transplantation and for the ear, for example, and looking over the horizon long term. Do you see regenerative medicine and bioprinting as being a sort of panacea in medicine, where we'll get to a point where if you have an issue with a certain tissue, organ, we'll just replace it?
I definitely think for certain organs,
Yeah.
that will be there. It'll be also interesting to see how some of like the uh, electrical implant work takes off with some of the BCI things and kind of
Yes.
from that perspective. Also, one thing I will point when you're talking about the interdisciplinary careers, I think it's funny because a lot of the original bioprinters came from like mainly mechanical and electrical engineers
Yeah.
because it's a mechatronic device, and that's why
Of course.
our group had a lot of successes because a lot of the initial biomaterials were very, very basic. And so we had a lot of materials expertise. But I just think it's funny. So you have a lot of the mechatronics stuff that, because I mean, it's essentially a printer, except you're just extruding, materials that are alive. And the other group that is also involved in, and also on like monitoring things is good software, having good computer scientists to
Yes.
monitor the quality of your print, quality of your cells. Interestingly, one of my students did a whole review paper on machine learning approaches, which I was kind of skeptical on, but
Hmm.
interestingly, with some of the bioink development, all of the ink components we use are really sophisticated, which means expensive. And so it's kind of interesting that he's been using machine learning to optimize our inks, and it actually does work and save money. I was kind of skeptical, and then I was like, oh, this is interesting to like use it to train. When you're looking at huge parameter sets, it's been really helpful.
So I'm glad you brought up BCI, so brain computer interfaces. There's obviously a lot of effort being done. In that area, the most famous would be Neuralink and what they're doing with their first patient there. Do you see a synergy between the sort of computer technology and being able to print neural tissue and that coming together, or are they mutually exclusive?
Oh no, no, definitely. There's definite overlap in application. I do
Mm.
have former students at Neuralink, so I should probably be careful about what I say. But I do know the field really well, and I do have my second book coming out in fall on the handbook on neural engineering. And so, interestingly, the same things that enable you to record from the brain are the same things that will enable you to monitor tissues, including our human tissue
Mm.
models. So I think that there's a lot of really good interplay there. Um,
Interesting.
and the same things that you'd want in a BCI, right? You want long-term electrodes, in this case, to either record, to receive and transmit signals.
Hmm.
Same thing would want to monitor our tissues. So there's definitely a lot of good synergy there. I did actually get a chance to visit Neuralink, and it's really interesting because outside of their BCI technology, and there's a lot of really good companies like BlackRock that
Yeah.
have made like really cool neural interfaces. Something that I was like really really impressed with at Neuralink was their robotic capabilities and the things that they're doing in terms of trying to make these things like consistently implantable using robots, which
Right on the surgery side.
on the surgery side, which I think is
Right.
really interesting. But yeah, there's definitely a big interplay, and actually, it goes towards the questions I brought up earlier. Is like, how would you know if your if your tissues you printed had thoughts? If we could. And I mean, people do recordings. As I said, the biggest issues are, you want you want the thinnest possible um, recording device to, whether it's
Right.
going in your brain or your tissue. because you don't want it to be destroyed. You want it to last, because that's a big challenge when you're implanting these electrodes in the brain is, they'll get glial scarring and then they'll stop working. So definitely something, and that's why I see a lot of interplay between the two of them is like, if you can record these signals, and it goes towards tissue regeneration. Way back in the day when I was doing my PhD, my PhD work was all focused on spinal cord injury, which we were looking at this from, can we replace these cells? And if we replace these cells, then can people walk again? And then also with Neuralink, it's like, well, if we could find a way to like detect, the brain saying walk, could we hook it up to, like an exoskeleton, which is like the other approach. And that also goes into like with BCIs and like it's interesting when like all the 3D printing stuff overlaps, but there's also, the huge interest in wearables and, can you detect, how people are moving and things like that. And then, you see a lot where, again, with injuries, you have a BCI and then, someone gets an exoskeleton for their arm. And so it senses when the brain thinks like, oh, my hand should be opening, and then sends it there. So I definitely think there's a lot of stuff that can all be overlapped there. And yeah, it gets really interesting, sort of with like the interfacing of, technology, whether we're using it, in our dish versus in humans. And it also goes towards like, again, if you're going to test your implants, and we've definitely talked to groups about that, and actually with the project we have with UBC, the Mend the Gap project, we actually print, although they don't have cells in them, we print these miniature spinal cords that mimic the spinal cord, and you can do injections of materials into them. So instead of having to use an animal, you can inject the material into our miniature spinal cords and they're clear. So you can see if the material is doing what it's supposed to do upon injection. And so
like diffusion studies to see where and how these materials diffuse?
Yeah, and also for the spinal cord injury, in this case, the materials are supposed to align. And so
I see.
It's easier to see if they're aligning in our printed ones before going to an animal. And so it helps cut down on animal studies. And that's tying that back to the BCI thing. Again, if you're trying to make the improved version of what Neuralink's doing, I think for those of you who followed, right, the initial patient had some of the electrodes come out because they were loose and things like that. It's a lot easier to test this in a bioprinted human tissue model, right, of a brain to sit there and say, okay, we've like had these electrodes in this, human brain model for 30 days, and then you can see what's failing, as opposed to when you actually have it in a person. So, and you can't do, you can't fully explore everything that's going wrong in that kind of system. And so I think that that's something that's really cool about 3D printing. And I don't know if I emphasize that on the drug testing part, or in this case, if you're using it as a test bed for recording electrodes, you do have a fully human system, which again, you shouldn't probably be having that much access to human brain tissue. But in this sense, like you can print a bunch of human brain tissue for testing, or skin testing for cosmetics and things like that, again, as an alternative to animals. So, yeah.
So is it safe to assume that companies like BlackRock and Neuralink are using your sort of technology?
I know they're definitely doing animal studies, and definitely are looking into, how, ways of looking at these things. And I do know, I believe Axoft out of Harvard has also been looking, at some of these, in vitro human things, because they're cheaper than doing, and also like things like monkey studies where you're doing very expensive things, and hopefully getting them right. And also with the pandemic, there's actually not that many primate facilities left. But it's definitely something I know that lots of work's being done around like long-term monitoring, and not just for neural, also for cardiac. How can we monitor these tissues, and what's the best way to get these electrodes in, and study whether it's these devices. And then I think it's a really cool technology transfer, right? Because if you can get really nice thin electrodes that can tell you what's going on in our tissue, it makes a lot of sense that then you could translate, it can go both ways with the tech.
So speaking of technology transfer, what is your opinion of bioprinted food? Lab-grown meat.
Yeah, I think it's interesting. I just don't think it's cost effective yet, because I think I have a lot of friends who do run, like I know Ali Khadim Husseini has Meat, and I know the like Stephen Gray, who's with New Bio out of Europe, and what they find is if you're going to grow a bunch of cells, and I think I've talked about this, whether they're human cells or cow cells, it's really expensive, and you need bioreactors. And honestly, I
Yes,
think their
that's right.
current uh, costly step is that a lot of these are grown in fetal bovine serum, uh, which is
Oh yeah, yeah,
taken taken from cow's blood, and so
yes.
that's, and as a person who runs a lab, is expensive.
Oh
And
yeah,
so if you want to have a lot
yeah,
of this fetal bovine serum to grow enough cells for your steak, I
yes,
think, right, I think like the burgers are like
yes.
$10,000 each. So they haven't gotten that yet. Although I do know, I think it's helped them come out with some more consistent alternatives to FBS. I think that's what you see in the meat cycle, is always like, here's our FBS alternative, and someday we'll sell food. So
Right.
I think those are some of the bigger challenges to overcome currently, is that I do know Dr. Akbari, who's at my university, was on sabbatical with Dr. Ali Khademhosseini's group, and he did, I think, eat their burger and said it tasted pretty good. So it doesn't sound like taste
Interesting.
is an issue, but I think the cost is one of the big things with that.
Yeah, there was a time, if I'm sure you remember, in 20, maybe 2015, 16, where 500 mils of FBS was about $500.
Yeah,
Or for some reason, not for some reason, there were good reasons that the price spiked. What does a bottle of FBS cost today?
I think it's around, yeah, like 300. It's still
Oh, really? Oh my
it's
goodness.
still like they haven't really, because with COVID, a lot of
Yes.
the animal derived stuff, like it took, like Matrigel was on back order for like years, and so people were looking at Matrigel alternatives. So a lot of the animal derived stuff, because so many people were trying to make vaccines and investing in biotech, there were a lot of shortages. So it's still, and then obviously now with inflation, things aren't cheap.
So, if you don't mind me asking a bit about your personal journey, how did you find yourself in Victoria? How did you find yourself in the sort of industry that you're in? Did you start off just as a bio undergrad, eventually made your way up?
Oh man. So I was always a nerd. So I always feel bad when students are confused about their life. Because when I was 5, you can find this, it's in a
Sure.
newspaper article. I asked for tickets to a Nebraska football game, keeping in mind I grew up in Missouri, so I'm a big sports fan, go Chiefs, and a chemistry set. And when I got my chemistry set, and apologies to all the parents listening, everything that said "don't mix this together," I like went in the backyard in Missouri. I was like the second of 3 kids, so this was the 80s, so it was wild. So I just would,
Oh, yeah.
just went and mixed everything they said not to mix, and some stuff exploded and some stuff turned pink. And I was so happy with my chemistry set. And so I was like, I'm going to be a scientist. And so did science fair all through junior high and high school. My high school senior thesis was on Dolly and cloning. So,
Oh,
and
okay.
I went to the local library at UMKC. Also really interesting in retrospect, now that I am a professor, and given that I grew up like not knowing professors, we wrote for my senior high school project, which was actually done for physics class. We wrote to a local professor at UMKC, and we did our senior physics project. Like, we took everybody's DNA, and I think we did PCR on it. And like, this professor,
Oh,
like, they were like, yeah, just collect samples. Like, you can come here, you can use our centrifuges and run gels and like write this all up. Which I was like, in retrospect, I was like, God bless that professor who was just like,
oh,
this high
yeah.
school kid, like this group of high school girls, they have a plan. And yeah,
That's fantastic.
went to MIT, majored in biology and chemical engineering because they didn't have bioengineering at the time, and that seemed to be what was really cool. They did have the bioengineering graduate program, so I was working in a bioengineering lab on tissue engineering, and started culturing cells, loved cells. Went to grad school at WashU. I worked with Dr. Shelley Sakiyama Elbert, and we did a bunch of neural tissue engineering, which back then was working with like mouse embryonic stem cells. Fell in love with the nervous system. As I said, my first love was spinal cord injury, so I did a lot of making tissues for spinal cord injury and things like that. Was a postdoc at Berkeley, and when I was applying for jobs, it was 2010, which was not a good time. So we were the only people who moved to Canada because the Canadian dollar was stronger than the US dollar, and we paid less
Yes,
taxes
yes,
in
yes.
Canada than California. And yeah, and then it was good. Yes, I started my own lab, and I wanted to focus mainly on human tissues. Got a Canada Research Chair and some other awards, and have been here since. And then, yeah, in the pandemic, another thing I don't recommend, we spun off axolotl. So that's kind of
Yes, I
how
was going to ask you about it.
yeah, it's kind of how I got from Missouri to Victoria.
So you've done sort of this jump that not every person that works in the sciences does, and that's to become an entrepreneur. So tell us about, because this is difficult, like you said, you don't recommend it, and I've heard that repeated many, many times. But I'm sure it's rewarding though when it works out. Tell us a little bit about Axolotl.
Yeah. Oh, I meant don't start a company during the week the pandemic shuts down
Oh, okay.
everything. Yeah. So we started working with Aspect in 2016, and we got their second ever bioprinter. And so we made this bio ink that worked really well with their printer. It won awards. It was like one of the methods of the year for ACS Biomaterial Science and Engineering. And we were like shipping our ink to a bunch of collaborators. And my co-founder of Axolotl, Laura, was my graduate student at the time, and she really wanted to start a company, and I was kind of indifferent. And in Canada, there's a lot of money to promote commercialization and protect IP and things like that. And so we were getting all these grants to commercialize our technology, but and since it's all worked out and Aspect is very successful now, so Aspect had sold like 7 printers at the time, and I think we were shipping ink to 3 of the 7. So every time we'd write a grant, we'd be like, we're going to sell our ink to Aspect's customers. It was kind of like, well, that's funny because we know all of them. And that's why they now do therapeutic tissues instead of selling printers. But so anyway, I was at the 2019 Biomaterial Society meeting, and Eric Gadom at the time was the CEO of Bico, who sells the selling printers, which they have thousands and thousands of printers installed worldwide. So we're drinking at a reception, and Eric's like, why don't we sell your ink? And I was like, oh, well, like we don't sell our ink because there's only like 7 Aspect printers. He's like, no, that's not what I asked. He's like, why doesn't Bico sell your ink? Because we actually have thousands of printers out there. And I was like, oh, well, that would be
Right.
like a business. And so, I came back, and then Laura was like, finally, you think we should do this? We incorporated, won some contests to get us some seed funding. And then, we incorporated the week of the pandemic. And then since then, we've been really lucky to get a lot of non-dilutive funding to help support Axolotl, and we've sold some products, done some consulting. And it's been interesting. I would say I've enjoyed it more than I thought. And also I think I actually gave like a talk on this probably a month ago at the World Microphysiological Society. It was an FDA panel on lab-on-a-chip devices and translating things. And there is actually a lot of benefit to thinking about commercialization of your products, or even just like towards reproducibility, because I was very guilty of this during my PhD, where I was like, you finally get your assay to work, and then you do that assay, and then you get your data,
That's
you
right.
publish your paper. And like for us with the bioink, we use it in our lab all the time, but it wasn't until we had a company that we were like, okay, well, like, how do we actually drive down the price on this? Like, what is the
Right.
exact minimum codes we need? How do we do QC? How do we make sure this is really reproducible, because we're going to be shipping it all over the world?
Yeah.
And like just that process alone, one, has saved the lab a lot of money, but also it's just like really good to improve the quality of your science. So that was something.
Yeah. It's optimization on a different level from a different perspective. It's an
Yeah.
excellent exercise.
Yeah. And that was why it was interesting, because when I was at this thing, and it's very interesting because there's like such a huge divide between like the academic microfluidic communities and then like the companies that make it. Because they're like, all these academics always start from scratch. And I'm like, well, they do, because PDMS is cheap. And so it was interesting to hear a lot of students. I'm like, yeah, you should go see what's out there commercially, and then, go back and apply that to your designs. So, but that's been, and then along the same lines, why I was giving that talk is we, for our drug releasing particles, we used to make them like you make salad dressing, so an emulsion, so you'd shake them up by hand. And then we switched to using microfluidics because it made them more consistent, but we were using PDMS, so you could run a little plastic chip for an hour. And then we got funding through the engine manufacturing supercluster to work with Starfish to make a commercial grade microsphere producer. And then that was what was really interesting, because then it went from PDMS to being all stainless steel and like really doing some intricate designs to upscale the production. And now we have a device you can just run constantly to make microspheres. And I was surprised because as an engineer, and with a company, we think about this all the time. But some of the students who worked on that project were from the chemistry department, and it was like their favorite thing, because they were like, finally, our work is getting out of lab, like it's getting like the industrial treatment. And it was really, I think, interesting for everyone. So I was like, that was really good experience, and just like showing how to go from something we made in a lab to having something to be commercial grade device was really an interesting process.
So this has been enlightening because in my mind, at least, having no experience with bioprinting, I thought it was a little bit of a niche industry, but it sounds to me like it's much bigger than what I anticipated. How far are we from, or I should say, yeah, how long away are we from seeing this sort of technology being used in every major hospital in the world?
I think we're starting to see it slowly, and I think you'll see it more in some of the major medical centers. Like as I said,
Mm,
in Vancouver, they've definitely already started planning to put this type of thing into their new hospitals. I'm sure places like Stanford are doing this. And honestly, how you'll see it for the clinical stuff, I think it'll be a core facility. One thing I didn't really touch on as
okay. Yep.
much is so one, like you'll need bioreactors to make all
Yep.
the cells to study. But it is an interesting skill set that you do need to run bioprinters because I have visited places where they'll have it's an interesting field. So you can buy you can for anyone on this you can Google like Hardware X, and you can actually retrofit something like an Ender to make an at-home bioprinter.
Uh,
So you can spend $300. I mean, you're not going to get good resolution. You're probably going to get super great cell viability, but for $300, you could home make a bioprinter. You can buy a BioX probably for around 500K, and then although Aspect doesn't sell anymore, you can get things like the bioassembly bot that you're looking at $250,000. And so I've gone to places where they've had some of these high-end bioprinters, and they just have no one who knows how to use them because it's
right.
such
Yeah.
you need to like know your CAD. You also need to know how to load your materials. You need to have, and this is where I think we really help out is with our materials is like doctors who want to bioprint tissues because they have all the iPSCs, they have the patient cells, and they just, want a quick way of making the same
Hmm.
tissues while keeping their cells alive. So it's definitely a really unique skill set. So I really feel like it'd be a core facility with trained technicians. And I will date myself, but like I always think of it like at med schools, how they have like a core knockout mice facility, where you have
Right.
trained
Yep.
technicians who know that because it's not a super easy skill to pick up. But I think that's where you'll start to see it's like at the major medical centers, and then the industry. It's been a bit interesting because several of the companies who I thought had really interesting tech that got bought by like traditional like 3D systems and stuff. They seem to have been getting cut with like the recent economic situation. So it'll be interesting to see kind of how some of them evolve. I do know United Therapeutics has a big interest in doing bioprinting, which interestingly they had done some printing, but it didn't actually have cells when they were trying to do their really complex lung models because
Mm,
again there's a trade-off between if you want cells or if you want resolution. And
right.
usually if you're trying to get high resolution you're using photocurable resins, which tend to kill cells the longer they're under the like if you're
Right.
using UV light then it's going to not be great for the cells. So it's definitely interesting to see sort of how the field shifts as like companies get bought and where different tech ends up.
So it seems like it's a growing field, and in light of what you mentioned regarding not enough people available to run these systems, that sort of thing, what sort of advice would you give to up and coming students who are, whether they're biologists, chemists, engineers, physicists, that are interested in this industry, in terms of they should study, or also what sort of skills and mindset sort of define people who excel in this area?
Yeah, I would definitely suggest, well, get your own 3D printer. It's in my
Mm,
office or my husband's office over there. We're a pair of engineers, so he has our 3D printer in his office. Just getting a 3D printer, they're like $300 to play around with. You get even cheaper ones is always good. I think CAD, and I know that I think you can get I think Fusion 360. I think students can get free copies of. So if you want to learn sort of CAD and how to make structures. I think that's one of the things that's really cool about 3D printing is you can make a design and then you can see if it prints, and it kind of teaches you some of the spatial things for that. Also, I think there's a really active community. I know Jenny Chen out of San Francisco runs 3D Heals, and they've got a lot of really cool webinars and events. And I think they're starting to do in-person events as well. Also, if you have local maker spaces, those are always good to check out as well. And then if you're an undergrad, I'd say go find labs to work in that have 3D printers. Is like they've definitely become more common. And I know at least at our school, UVic, we do have a 3D bioprinting course that I started, so it does give you some hands-on experience. So there's definitely coursework out there, and I believe it's interesting because Australia and New Zealand have really active, and also a lot of Asia does. I just got I was in Korea in May, and they have a lot of really interesting bioprinting companies in Korea. But I think that one of the New Zealand universities has like a free online course on bioprinting. So those would just be some of the resources to look into.
Now, I've got one more question for you. I've got a couple of questions, but how much of this technology on the CAD side and the software side is open source? Is there a push for that, or like with most pharmaceutical industry, proprietariness is kind of key?
That's really interesting because for most of the designs we bioprint, just given that they aren't as, you aren't getting the same level of resolution you get with traditional 3D printing. Usually we're printing
Yeah.
relatively simple structures. One of the things that drives me nuts is you'll see a lot in 3D printing papers is they'll print a grid. And I'm like, there is no grids in our body. Like, do not print a
Yeah.
grid. Which I said at a McMaster symposium, and everyone was very, very excited. But people like to print grids because then you can
Somebody
see.
finally said it.
Yeah, they're like, well, because you can see how like even your lines are. You can say, okay, we achieved this
Right.
resolution, but there's no grid in our body. So we usually print, as I said, like half domes. We actually have some more complicated designs that come preloaded on the cell ink, but sometimes you get into some interesting constraints when you're printing like brains with the actual wrinkles on them and things. So there is actually, interestingly, usually most 3D bioprinters come with their own homemade CAD software that's relatively simple because you're usually printing stuff like rings or half domes or rectangles. And then you can also, if you're more familiar with like SolidWorks or Fusion 360, you can go download your STL files. There's actually a ton of those available for free on the internet, like tons of repositories. I taught 3D bioprinting. I also taught a regular 3D printing class, and sometimes they would find stuff, and I'm like, "Where did you get this?" And they're like, "Oh yeah, like online. I found this STL file of a bone scan of someone's broken leg," and I was like, "Oh, okay."
On GitHub or something.
Yeah, on GitHub. Yeah. So I think there's definitely things out there, and I'm sure there are open source CAD programs you can use. But I find in industry they're either using SolidWorks or Fusion 360.
Are you worried that, for example, I mean, there's some worry with biohacking in terms of cloning and subcloning has become super easy. You can literally order kits or they're doing it in elementary schools nowadays. Are you worried that this might become an issue in the future? Printing tissue at home and going awry?
I mean, possibly, but usually if people are doing that, it's not like I'm a professional engineer, so I'm a licensed professional. Like it's usually doctors with ethics would not be doing that. I mean, it's definitely like there's definitely been 3D printing stuff implemented in countries where they don't have the same level of health oversight. So I think it is a bit concerning, but those studies haven't been anything, major like transplanting a heart. It's been more, this is a potential wound substitute and that kind of thing. So I think we're ways away from that. And I also think people would be slightly skeptical, though I will say I was like very shocked when the doctor in China actually did CRISPR people, and that happened.
Yeah.
So
yeah, and admitted it. Yeah.
That was one of those moments because in one of the classes I would teach, I'd be like, yeah, you would never CRISPR humans. And then someone did. So then now I have to always be like, you really shouldn't do this. So
That's right. So what is, wrapping up, what is your hope in terms of where this technology is 10 years from now?
Oh, yeah, I definitely hope it enables more personalized medicine and better treatments. I'm very excited with our collaboration where we're actually working with patients who have Alzheimer's, because in that case, like working with MD PhD, we could potentially see drugs back in patients within a year or two. And I think that is one of the most exciting projects with the patient-derived cells where you can have an impact, although it's on a much smaller scale. Although if something does work, it could, my collaborator could use that to eventually drive forward a clinical trial. The same with some of the cancer work. I'm very excited to see what happens with the team at Aspect, having known them for a long time and having worked with them. It's really exciting to see their success. And it's also been really exciting to see Canada invest both in them, and also we've been really lucky at Axolotl to get a lot of support from the government and also from British Columbia, which does have a biomanufacturing strategy. They just released their progress report. You can see Axolotl photos on the cover. For our group, yeah, it'll be great to do some more modeling of Alzheimer's and Parkinson's. And one thing I hadn't really touched on, but with our collaborator, it's interesting with Alzheimer's, and as you derive these patient-specific lines and develop them into models, is Alzheimer's is a lot like cancer, whereas like the most easily studied forms of it that are inherited are a very small portion of what is actually classified as the disease. And so it's been really
Right.
interesting with the patient-derived lines to see the variability. And something that we are looking at is, for the people with the same type of mutation, which should be the same type of Alzheimer's, like how robust is the phenotype between different patients? Like, what would we call? This is something we've talked. I talked with Jackson Labs, who sells all the animal models of Alzheimer's, and something we talk about is like, so what does a very robust human tissue that would replace a mouse look like? Because I always say with the Alzheimer's work, and this is, I guess now very applicable, as my parents get older, if it's my dad with dementia and it's his own cells, and this drug works on a tissue that's made of his own cells, definitely give it to him, but don't give it to everybody's
Right.
grandpa, dad. But like, what would that be like? And so we've talked with Jackson Labs about comparators of the Alzheimer's mutation in their animals and our human iPSC lines, some of which we get from Jackson Labs, like to do the comparator of what does the Alzheimer's look like in our bioprinted brain versus the actual animal. And something that's really interesting that we could add in with them, because they are the mice, you could actually print the mouse brain with the exact same genetics and see how similar the printed brain tissue is. I think that's something you're getting at earlier, to the actual mouse brain itself, which I think would be super
Yeah.
interesting to do the across the board. That with also the across the board drug screen would be really cool to see what correlates and what doesn't.
I've always thought of personalized medicine from the genetic aspect. You do a screen, you do a chip of, genes on a chip and screen for what you may or may not be predisposed to, and then come up with a plan from there. But I've never thought about it in terms of doing personalized disease models. So that's fascinating. I thank you for illuminating that for me. Fascinating work. Dr. Stephanie Willerth at UVic. Tell us a little bit about where we can find more information about your lab. If there's any prospective students that want to contact you, how do they do that?
Yeah. So I think if you just Google Stephanie Willerth, the lab website's the first one that comes up. I'm also on LinkedIn. So if you want to find out what we're up to, Stephanie Willerth on LinkedIn. I'm also Dr. Willerth on Twitter. I think those are pretty much the main ways to find me. And if you're ever in the area, drop me a line. We always are happy to give tours to people to show our bioprinters off.
All right. Thank you so much. I appreciate it. It was a pleasure.
Awesome. Thank you so much.